Okay, I see a few more names appearing in the attendee list. Hey, hello everyone. I see many names trickling into the list. It's so nice to have you all here with us. If the CART transcriber is here, if you wouldn't mind letting me know, perhaps in the chat. I cannot tell by the names and I don't see your name in the participant list either. For everyone's information, we want to ensure we have full access to the presentation. So we are currently waiting for the CART service provider to arrive. I think we'll give it another minute or two. And if they haven't joined us by then, we can begin the presentation with the automated captions. And once the captioner joins, I will share the link with everyone. Okay, it seems they have arrived. I am trying to figure out the technological aspect of this. My apologies. So I have assigned panelist role to the captioner. I'm not sure if it's working. Oh, yes. Okay, it seems to be working now. Okay, we do have the captioner here with us now. If the transcriber is able to send me the link, I can share that with the group as well. My apologies for all the technology issues. It's our first presentation of the academic year, still adjusting. Okay, I'd like to begin by saying thank you everyone for joining us today. We are truly honored to have you here as well as our esteemed presenter. This year's distinguished lecture series will begin with today's topic on neuroscience, language, creativity, emotion, and how the four modulate learning. The intent of our presentation series is to bring in world-known, well-reputable, distinguished presenters to share their research, their findings, and just all of their amazing science. We are honored to have our first presenter of this year's series, Dr. Vonetta Dotson. Dr. Dotson has also contributed to our diversity, equity, and inclusion in science presentation. This year.
Ella, your sound. Maybe check your sound. Apologies about that. We'll have the other interpreter check their sound.
Is that better?
Yes. Mm-hmm. Oh, go ahead.
So this year we will be emphasizing those presentations that contribute to equity, diversity, and inclusion as areas of focus. Gallaudet as a university has been focused on providing more equitable and diverse collaborations for our students. In the chat box, I will be posting a link to the list of this year's presenters, as well as sharing information on how to sign up for our VL2 listserv. From there, you can find various archives, videos, further information on our page, including archived footage of past presentations. And in the same location is where you will find all current and future recordings. Now, before I turn it over to our students for a formal introduction, I'd like to just briefly walk through communication processes to let you all know. Today we will have the chat box enabled. It is more for the purpose of if you are having any difficulties visually accessing the content or the interpreter, any issues with the audio or the webinar, technology difficulties can be shared through the chat box. If you have any questions for our presenter, I do ask that you submit those in the Q&A feature as opposed to the chat box. All questions submitted to the Q&A will be held until the culmination of the presentation, at which point we will read through those questions and answer them. That, I believe, leads to a bit of a more efficient process. I think that's all for me. So I'd like to turn it over to our students, first beginning with Rachel Sortino, who will announce herself as well, and then formally introduce our presenter. Hi, I'm Rachel Sortino. This is my sign name. I'm a first-year Penn student. Welcome everyone to the PhD in Educational Neuroscience Distinguished Lecture Series hosted by Gallaudet University and the first of this year. The goal of this series is to honor world-renowned scientists in a variety of expertises and fields, including psychology, education, cognitive neuroscience, neuroscience, and a number of other cross-disciplinary fields as well. Their contributions to the field of educational neuroscience allows us to grow as an ever-expanding field and deepen our understanding of the human mind, as well as how we as humans learn. I'm honored to introduce to you our esteemed and quite distinguished presenter, Dr. Vonetta Dotson. Dr. Dotson is a professor for psychology and gerontology. And is the founder and president of CerebroFit, Integrated Brain Health. as well as a senior scientist for NASA. Dr. Dotson's area of expertise and study focus on positive and negative modifiers of brain health, including how depression, cognitive, and the aging process all align and interact with one another, how disparities may impact brain health, and how exercise may be used to disrupt any potential cognitive, brain, and emotional functions. Her company, CerebroFit, provides empowerment for people in their lifestyles for brain health and improvement. She has published a book entitled Keep Your Wits About You: The Science of Brain Maintenance While Aging. Dr. Dotson has been selected as a fellow for the American Psychological Association's Society for Clinical Neuropsychologists. Today's topic will focus on vulnerability in underrepresented vascular depression, expressing a need for diversity in clinical neuroscience research. I'm so happy you're able to join us today, Dr. Dotson. I will turn the floor over to you.
Thank you so much. It's my honor to be here. I really enjoyed having a chance to meet with some of the faculty and students earlier, and now I'm really excited to share some of the things that I'm most passionate about. Let me go ahead and share my screen. Okay, I'm going to assume you can see it. And please let me know if you cannot see the slides. So as you heard, I wear a lot of different hats, work for a lot of different organizations, but everything I do is under the umbrella of promoting brain health. And when there are things that get in the way of brain health, trying to figure out how we can overcome that. That's why I'm so passionate about increasing diversity in the research that we do to make sure that we're able to really help everyone to benefit from the science that we are doing that informs our clinical care. And my particular interest is in vascular depression, as you heard. So one reason why I focus on vascular depression, which is a subtype of depression in older adults, is that the population is aging. We're going to have an increasing number of older adults in society who really are in need of clinical services. In addition to that, the racial and ethnic diversity within older adults is going to continue to grow. So the figure on the left side just shows you sort of the makeup, the ethnic makeup of the population that's estimated for 2045. As you can see, only about half is non-Hispanic white. The other half is made up of other ethnic and racial groups. And we know that there are differences in access to healthcare, risk for diseases, and other important factors in different groups. So we really want to be mindful of that. And when we think about aging in particular, you can see that projections for 2050 is that non-Hispanic white individuals, the proportion will change and get lower, but for all other ethnic and racial groups, the percentage of the population will continue to grow. So that means that we really need to make sure that as we do research, we are including people who represent different groups. For this presentation, I'm going to focus on racial diversity because most of my research on health disparities is in Black older adults, but this applies to all aspects of diversity. That can be socioeconomic status, native language, sexual orientation, disability, etc. These are all variations that people have in their identities and who they are that can impact so many things that we research, and all of it should be, all aspects of diversity are important. So I do not at all mean to minimize that, even though my focus will be on this population of Black older adults. So related to what I said a moment ago, because I mentioned lots of different aspects of diversity, there's this idea of intersectionality. So we know that when you have multiple aspects of diversity in the same person, that can present a unique context and unique challenges and opportunities because of their multiple identities. So for the work that I'm doing, I think in particular about the intersection of race and aging. So for example, the current cohort of Black older adults grew up during the civil rights era. They grew up during a lot of the things that we currently read about, and it seems like for our history, and that was their reality. My own mother talks about, not being able to go to certain playgrounds when she was a kid. And she lived through that. That affects her perspective on the world. And that's going to be different than people who are our ages now, who then become older adults, who live through different times. We've lived through a pandemic. We've lived through the different things that happened in 2020. So every cohort has differences. And so the age and race right now has a very unique context compared to how it would at any other time in history. So that's an aspect of intersectionality. You have race, you have age, but you also have time and history. And those contexts are really important to keep in mind in all the things that we do. So when we think about intersectionality, I like to think about things from a biopsychosocial context, so biological, psychological, and social factors. This is something that we do a lot in psychology and neuroscience, so many of you are probably familiar with this idea. I wanted to give some examples of intersectionality, knowing that this is not comprehensive, but just wanted to give some examples. I have within each of these ovals, these sort of darker gray or black letters are known race-related risk factors. And then in the beige color or white color are more age-related risks. These can overlap, but I wanted to show how if you have someone who is, for example, a Black older adult, they're having this combination of risks. So for example, on the biological side, as we get older, but also based on racial differences and disparities in healthcare, chronic medical conditions are going to be a greater risk. Exposure to pollution and other toxins might be greater in racial and ethnic minorities. Access to good nutrition might be different. So that's on one side, you have the race-related risks. But then when you have someone who's also older, you might bring in things like hearing and vision changes or brain changes that are also going to be a part of the picture. So you can see from that one example how you're having this combination of different risk factors coming from different aspects of one's identity. And those things can create a unique context. The same thing is true for psychological risks and for social risks. So I know at least some of you have had a chance to read the article from a couple of years ago that really talks about how the neuroscience research that has been out there historically really has not kept up with how the demographics in our country are changing. One thing I want to point out is that this paper was published in 2020. We did our examination of articles in different journals from 2018 to 2019. And I say that because I do think that some of these numbers would be better if we look now, because it seems like there's more understanding and acknowledgement that we need to be more transparent when it comes to reporting different aspects of diversity and in trying to include more diverse samples. Does that mean that I think that everything has been solved? No, I do not. We have a long way to go, but I did want to give that acknowledgement that it would be interesting to see if the numbers were quite as bad if I were to do them, let's say from 2021 to 2022. But having said that, as a brief summary of what we found, we looked at 2 of the top cognitive and affective neuroscience journals in the field and looked at every article that was published for a 1-year period in those journals. I'm sorry, for a 2-year period. And we calculated how often these studies reported certain demographic factors, such as race and socioeconomic status, or SES. And then out of those that did report race, we looked at what was the percentage of different groups in their samples. And as you can see, the numbers were not good. Only 14% even reported race or ethnicity. I found that really shocking. It just seems like such a basic thing to report in your sample characteristics table, but only 14% did. 18% reported SES, and less than 1% actually looked at either race or SES as a predictor, meaning that they were not interested in seeing, are there differences across or between different races or people of different socioeconomic status. Also disappointing to see was just the number of, or the lack of people from different ethnic and racial backgrounds in the studies that did report them. So you can see that there was at best 50% non-Hispanic white in studies, so ranging from 2 to 50% Asian participants. I will say I should probably put individual numbers in here because it looks deceptive to show 50% because only one study was that high. Most of them were under 20%. But there was one that had 50%. And then you could see as few as 0 and then up to, in one situation, 42%, either Hispanic or African-American. So these numbers to me are completely unacceptable. We know that we want our research to represent the populations who are going to be affected by that research. We want the healthcare that we give to be informed by research that is representative, and this is not at all representative. So I would like to use vascular depression as an example of just how much this lack of diversity contributes to health disparities that we see. So we know that Black older adults are at a greater risk for depression compared to white older adults. And we also know that the severity of depressive symptoms tends to be higher in Black compared to white older adults. On top of that, in older adults, we often see a higher prevalence of what is sometimes called subthreshold depressive symptoms. That means you have symptoms that don't meet criteria for major depression, but they're still clinically significant because they still affect functioning, they still affect cognition, they still affect the brain. So in general, older adults tend to have more subthreshold symptoms than clinical depression, but the prevalence of subthreshold symptoms is higher in Black older adults than in white older adults. What's also really important is that depression seems to have a more negative effect among Black older adults, meaning that the effects on, social functioning, work functioning, everyday functioning, all of that is going to be greater in general in Black older adults compared to white older adults. So there's a vulnerability there that's very important to acknowledge. So I would like to switch and talk a little bit about the brain basis of depression, because this will explain a little bit about vascular depression and what exactly that is. I know some of you had a chance to read the review paper that I wrote with my graduate student. I give her the credit for doing most of the work on that. I am blessed to have a really fantastic lab, and they're all very bright, motivated students. So depression, we know, has a basis in the brain. We know that there is a network of regions in the brain that need to operate and function well together to maintain our mood. If there is any type of disruption to those networks, either one of the regions themselves or the connections between those regions, then depression can be one of the consequences. So we call that network, you'll oftentimes see the term frontal limbic. So frontal limbic regions or frontal limbic network. That's the sort of collection of brain regions I'm talking about. What we see in depression are a number of structural and functional changes. Usually we see decreases in volume. We have decreases in cortical thickness. Blood flow is altered in a way that is detrimental to cognitive functioning. Sometimes that can mean too much blood flow in some areas and too little in others. And functional connectivity changes. So that means when more than one region operates at the same time, they're functionally connected, and those patterns of connections can be disrupted in depression. What's important for my interest in vascular depression is that we have a lot of changes in white matter. And that's important because vascular diseases tend to affect the white matter in the brain. So I'm sure you all know, because you're a neuroscience department, those are going to be your myelinated axons, very important for connecting regions of the brain together. And we see lesions. Was someone trying to stop?
I just needed the myelin, that word you said. If you could go back to that one spot, please. Thank you.
Okay, yes. So white matter is the myelinated axons in the brain. So it's the part of the neuron or brain cells that is connecting, each brain cells to other brain cells. And again, I know I'm saying this to people who probably know this just as well or better than me, because you all are studying neuroscience. So lesions within the white matter can be caused by vascular changes, and other white matter changes include a decrease in the integrity of the white matter. Other changes that can happen with depression, we have an increase in inflammation. So we know that inflammation is a natural part of the immune system, immune reaction. But when you have chronic inflammation, that can cause problems. And we see that in depression, the state of chronic inflammation. We see increases in cortisol, which we know is tied to stress. And then we see decreases in BDNF, which is a chemical we know is important for the health and survival of neurons. So these are this is a summary of changes that we see in the brain of people who are depressed. This can be true for depression at any age. It is particularly true for people who are older. The older we are, the stronger link we see between brain changes and depression. So this figure that I have here is showing you how many, I may have missed a message about stopping. I can't see the chat, my bad.
Good to go, Dr. Dotson, thank you.
No problem. The figure shows you how many of the changes that happen in the body and in the brain related to depression are shared with cardiovascular disease. And I'm not going to go through those in detail, but I want to say that as a transition to talk about vascular depression, because it's important to know that there are biological links between depression and cardiovascular disease. So this takes us to the vascular depression hypothesis. The idea of this is that vascular diseases can worsen or bring about a case of depression or an episode of depression. So having chronic vascular disease can make you more likely to become depressed. If you have recurrent depression, it can precipitate an episode. For example, if you have a stroke or a heart attack. And it can also prolong episodes of depression if you have chronic vascular disease. The idea is that this is because chronic vascular disease eventually disrupts those frontal limbic or frontal subcortical networks. And so the white matter lesions that I mentioned in a previous slide are really what we see as the cause of vascular depression. So those white matter lesions interrupt the networks and then lead to mood dysregulation, mood problems. And what we see is that people who have this cause for depression tend to have distinct clinical symptoms. So they might have more executive dysfunction, more psychomotor slowing. Oftentimes we'll have more anhedonia, meaning that loss of interest or experiences of pleasure. Maybe we'll have less sad mood, but more of that anhedonia. And we see a lot of functional disability that can be worse in people who have vascular depression compared to depression that doesn't seem to be tied to any vascular changes. There's some evidence that as many as 50% of older adults who have depression have a vascular basis for their depression. So this figure is just showing you some of the different mechanisms that could lead to vascular depression. And so as you can see, there are vascular risk factors like high blood pressure or high cholesterol that can ultimately lead to lesions in the brain, in particular those white matter lesions. And that can be exacerbated by things in the environment. So negative life events, insufficient social support, all of that can contribute to it and bring about a vascular depression. It's a good example of how the environment and the biological mechanisms interact together to lead to mental health problems. Because in this case, we're saying that on a biological level, we have these changes in the vascular system, but they're being exacerbated by things going on in the environment. So there are a number of studies that support this idea of vascular depression. So for example, we know that the burden of white matter hyperintensities or white matter lesions tends to be greater in depressed older adults. I'm sorry, when people, scratch that, sorry. People who are depressed and have cardiovascular risk factors tend to have higher white matter lesion burden and more neuropsychological dysfunction. We also know that people who have more white matter lesion burden and more severe depressive symptoms have a worse response to antidepressant treatment. So it suggests that vascular depression is particularly hard to treat. When people have progression of their white matter hyperintensities and then also have cognitive decline, then that tends to predict a worse course of depression. So you have this interaction between the white matter lesions, the cognitive decline, and the depression. And then there are a number of studies, functional neuroimaging studies, that show that you have basically altered prefrontal connections to other parts of the brain. And that tends to be associated with white matter hyperintensities. So this slide shows you some data from studies that I've done in the past. These were actually done when I was a postdoctoral fellow, using data from the Baltimore longitudinal study of aging. So this study's been going on since the 50s, actually, and then neuroimaging was added in the 90s. And there's a lot of data, annual data about people's depressive symptoms. And then also since the late 90s, annual MRI scans. So for this sub-study that I worked on, there's actually two of them represented in this figure. On the left side is a study where we looked at white matter hyperintensities. So there were ratings of how severe the white matter disease was. And we looked at changes over 4 years in those white matter ratings. So in this case, we don't want white matter hyperintensities. So bigger is not good. And you can see that the darker the bars on this figure on the left side, the higher the depressed mood scores on a questionnaire. And so you can see that as the depressed mood scores got higher, the changes in white matter hyperintensities over 4 years was greater. And that's especially true at the oldest ages. So the 3 sets of bars represents different age ranges, and the people of the oldest age ranges had a really huge effect. So there's this age by depression interaction, where the older you are, the more of an effect you're having of the depression. The 2 figures on the right side are from a related but separate study where we looked at changes in the volume of white matter lesions over 5 years. And this time what we saw is that for symptoms of depressed mood, as well as somatic symptoms of depression, so that means things like loss of appetite or low energy, each of those showed a relationship with white matter lesion volume. So the higher the depressive symptoms over time, the more changes, the more increases in white matter lesions over 5 years. So this is just showing you some supporting evidence for the vascular depression hypothesis. So I want to show you some data that is hot off the presses. I am working on a paper with a colleague of mine at University of Florida to look at trying to understand more about the effect of white matter hyperintensities as a measure of vascular disease in Black older adults in particular. So what we did, and it's one of the things that I recommended in that review article about the need for diversity in cognitive neuroscience, there are a lot of publicly available large data sets that might include thousands of people that have been collected. And as researchers, we can put in a request and get that data. And in the past, I've always thought, oh, these studies are not very diverse. They only have maybe 5 or 10% of either Black or Hispanic or other groups. But when you think about it, if there's 10% of a sample of 2,000 or 3,000 that is Black, that's still enough people to do some analyses, to be able to have some targeted questions that we ask to understand how things might be different or possibly the same in different racial groups. And so that's what we did. We got a subsample of Black participants in one of the publicly available data sets. And we looked at whether or not depression and white matter hyperintensities interacted to increase dementia risk. So this figure is kind of complicated. I'll try to break it down. The solid bars represents the analysis of people who had depression in the past month. These kind of dotted bars or speckled bars is looking at the analysis of depression in the past 2 years. So everyone was asked, have you been depressed in the past month? Have you been depressed in the past 2 years? Yes or no question. We wanted to see if having been depressed, either in the past month or past 2 years, combined with having high white matter hyperintensities, if that would increase the risk for dementia. Now, when you think about it, because vascular depression is the combination of a depressive disorder and a high white matter hyperintensity burden, that means that we're kind of looking at vascular depression, even though we're not calling it that. And I say that for a reason. If you look at these different sets of bars, the first set represents people who did not report being depressed at any time point, and they had a low burden of white matter hyperintensities. The second bar or set of bars represents people who had been depressed, but didn't have very many white matter hyperintensities. So you can think of that second set of bars as depression that's not vascular depression. So it's late life depression, not having the vascular part of it. The third set of bars are people who did not report depression, but did have a high burden of white matter hyperintensities. And then the last set is really getting at vascular depression. These are people who reported depression and also had high white matter hyperintensities. What you can see is that top darker section of the bar represents the percentage of people who had dementia. And so if you look, this is one of those things where you don't even need statistics. You can just look and see this huge effect that the people who had depression and high white matter hyperintensities had a huge risk of dementia. For people who had depression in the past month, plus high white matter hyperintensities, 25% had dementia as well. For people who had depression in the past 2 years, plus the high white matter hyperintensities, 40%, huge, huge number. And that's going to be in contrast to all the other groups that had anywhere between 4.5% and 12.5% risk for dementia. So this really shows you how detrimental that combination of vascular disease and depression is. And it's showing you in a group that is very vulnerable, because we know that there are health disparities in vascular diseases. So that means that the risk for depression and dementia is much higher in a very vulnerable group.
And we're going to switch interpreters at this slide. Just 1 second.
So as I mentioned, we know that there is a racial disparity in vascular diseases. This has been shown for many different conditions that affect the heart or just the general blood supply, things like stroke and arterial disease. We also know that based on the general literature and the slide I just showed you, that having vascular risk factors can increase the risk of depression and cognitive changes in older adults. I should say specifically in Black older adults. So it's important to know that there's an increased vulnerability to vascular depression that can be heightened by race-related stress. And this is one of those ways that we have extra risk factors based on intersectionality. Someone who is older and also from a racial minority background. We know that, again, Black older adults have gone through a period of history where there were even greater limitations in the healthcare and housing and employment, and that some of those things do continue today. We know that that cumulative experience kind of adds up in terms of chronic stress. Those limited access to resources can create barriers to having healthy aging and having more productive aging. And we know that this can contribute to different health disparities for things like depression, anxiety, and dementia. The research shows that discrimination of any sort, and this can be due to race or any other factor that a person might be discriminated against, has negative effects on cognitive functioning. And again, we know these health disparities are not just race-based, but also based on gender, socioeconomic status, and other factors as well. And many studies have shown that chronic discrimination can lead to health problems. Everything ranging from breast cancer, high blood pressure, and even risk for death can be greater as a consequence of facing chronic discrimination. And I think it's important to know that this is true regardless of, I hate to say severity, but for lack of a better word, severity of the discrimination. And what I mean by that is that people will refer to, what we call microaggressions. I always say that I don't like that term because micro implies small, and these are not small things. If someone is having sarcastic comments made or being, treated differently or looked at differently when they go into a store or go to a restaurant, those are things that add up. They get internalized. And so they're not small. And so the research shows that it's not just things like not being given a job or having someone, verbally attack you for your identity. It's also those so-called microaggressions that can be associated with a lot of negative health outcomes. The research shows that when we have chronic discrimination, our body responds. Our body shows, kind of carries that discrimination. We have an elevated stress response. So we're feeling this sense of being attacked or being worried about facing discrimination that can cause hypervigilance. So looking for negative things to happen. And for the body, that's going to show up in cortisol levels being higher. We know that when cortisol stays elevated, we can lose neurons in the hippocampus. We know that can change the process of brain myelination and lead to a number of changes in the brain, such as overproduction of myelinating cells. We know that overall this can start to contribute to more permanent types of brain damage. And that puts a person at risk for different disorders, cognitive disorders and mental disorders. So many of the health disparities that we see in different marginalized groups can be tied to the effects of discrimination on the brain. I'm going to skip this for the sake of time because I know that we got started a little late. I want to go back to vascular depression. I mentioned what I did about race-related stress because that is likely one of the reasons why we not only have racial disparity in risk for vascular diseases in general, but also one would guess that we would have a racial disparity in vascular depression. Despite that likelihood, there are very few studies that have actually tried to look at this. So I mentioned the study that my student Hannah conducted, which is one of the studies that was sent out for you all to read, try to see what's out there in the literature that talks about vascular depression in Black older adults. So as she was doing the study, she initially did a lit search that was, I think she looked in quotation marks, she looked for vascular depression and then Black older adults, and only found 3 or 4 matches. And then when you read the abstract, they weren't what she thought they were. And then realized that there were only like a handful of studies that truly looked at vascular depression that had a sample of predominantly or a good proportion of Black older adults. So she had to lower her standards basically to say, okay, let's look at any study that has either at least 75% of the sample that was Black, or if they had less than that, how many of them, or which studies looked at race-specific analysis. So they used race as a predictor. And so after using that more lenient criteria, she was able to identify 30 studies that sort of looked in general at vascular conditions and depression, but most of them didn't actually call it vascular depression, only 2 did. And that's important because how we operationalize or how we try to define vascular depression using best practices needs to be applied to the people who are most vulnerable. And prior to the work that we're doing in my lab, only 2 studies have done that in Black older adults. So you can see some of the characteristics of those studies here. And importantly, only 1 study used imaging, only 1 study used MRI. That's important because the best way to define vascular depression is to see imaging evidence of white matter hyperintensities combined with a depression diagnosis. The studies that didn't use imaging based it on self-report or medical records indicating multiple vascular conditions. That's fine if you don't have imaging, but it's not the ideal way. So best practices for doing vascular depression research had only been done in 1 study ever in Black older adults. Again, until the work that I'm very proud that we're doing in my lab right now. So from putting together the information from studies that were related, but not exactly the same as vascular depression, that at least gave us some clues for kind of where to go with this research next. She did find that vascular conditions were associated with depression risk. So the more severe different vascular risk factors were, the more likely people were to have depression in these groups that were predominantly Black. That having a vascular disease plus depression was associated with cognitive problems, functional disability, and other negative psychosocial outcomes. In her systematic review, the data on race differences was mixed, like whether or not there was a higher prevalence in Black older adults. But I think it's really important to remember what I just said, which is that only 2 studies called themselves vascular depression and only 1 used imaging. So it's hard to really know about the prevalence of a condition if you don't actually define it appropriately. So I think the jury is still out on that. So as I mentioned, only 1 study included neuroimaging, and that was a small study. And this is not trying to knock the researchers, because I'm really glad they did this, but it just points to the fact that we need more of this. So in a study of 46 people over the age of 50, they did use MRI scans to define vascular depression, which is great. This study did find that the rate of vascular depression was higher in Black compared to white participants. They also found that there were other risk factors, such as being female, having hypertension, or motor slowing, having more executive dysfunction on neuropsychological testing. Interestingly, the people with vascular depression had a lower rate of family history of depression. And that fits with other research in late life depression that shows that as you get older, your risk for depression is more tied to changes in your brain and less to family history. So it's kind of like the genes are less contributing to depression as you're older. And instead, what's most important is what's happening in your brain. And a big part of what's happening in your brain is what's going on with your vascular system. So this means that, what I summarized is that Black older adults are really poorly represented in research on a condition that they're vulnerable to. And that's really, I think, a way that research is not doing what it should be doing to address health disparities. If we know that certain conditions are more common in certain age groups, in certain races, in men versus women, then we all know it just makes sense to make sure we do plenty of research in those groups who are most at risk. And that's not happening in vascular depression research, and that really needs to change. This is particularly troubling because vascular depression seems to have a lot more negative outcomes than depression that's not due to vascular disease. So there's evidence that there's more cognitive problems, there's more functional disability, and there's worse treatment outcomes in vascular depression compared to depression that's non-vascular, if you will. So again, this is a really bad type of depression. We have a group that's very vulnerable, but not being represented in the research. So I'll pause here. So there's multiple implications for this. I think it highlights for us that the research that we are doing might not be able to be generalized to the populations that are most at risk. And we want our research to be representative. That's just a basic tenet of research, that we want to increase generalizability. And so lack of diversity in the research limits that generalizability. I think when we don't have that diversity in research, we miss out on opportunities to be able to personalize our treatments. So since I am a clinical psychologist by training, I'm licensed in the state of Georgia. What I do through my business, CerebroFit, is actually assessment and therapy for older adults. So I always have that clinical hat on. I'm always thinking about how is my research going to help me and other clinicians better treat our patients. And one way that we can treat our patients better is by personalizing our interventions. And what I mean by that is that if we know that depending on the person's demographics, their gender, their age, their race, they are more at risk for certain conditions, or if the basis for their depression might be different, right? Someone who's older is more likely to have a vascular basis for the depression than someone who's in their 20s. So how I treat the depression in older adults might need to be tailored. I might be thinking more about addressing the vascular basis of the depression, where I'm not going to go that same route with someone who's younger and has different mechanisms at play. So personalizing based on the person's characteristics, based on their clinical presentation. A lot of my research, which I didn't get into for this talk, has to do with understanding the variability within depression. So without getting into a super long discussion about that, to get a diagnosis of depression, we have a group of different symptoms that a person can report. Things like sad mood, loss of interest or pleasure, sleep problems, appetite problems, etc. And you don't have to have all of those symptoms to get the diagnosis. So what that means is I can have 10 people who have depression who have completely different symptom profiles. Their actual symptomology, their symptoms might look completely different. Well, what the research shows, a lot of work in my lab, a lot of work from other people, is that those different symptoms seem to have different biological contributors. So some symptoms are tied more to changes in one brain region or another. Some symptoms are tied more to inflammation versus vascular changes versus neurodegeneration. It varies a lot. So what that means is that if we paid attention to the symptom profile of the person, maybe we would realize that certain treatments that target what seems to be the underlying biological cause might be more effective. Again, I could spend all day talking about that because it's something I feel really strongly about as well. I have a paper that my lab wrote on exercise as an intervention for depression in older adults, and in particular for cognitive impairment. So it's a review paper that kind of puts this all together in a way. I'm happy to share that if anyone is interested in reading a little bit more about this idea of targeting the underlying biological mechanism in depression treatment. So I will stop with that tangent and come back to the slides. Another implication is that we really are perpetuating health disparities. Inaction is still problematic. When we think about health disparities, even if we're not overtly trying to keep things unequal, even if we're not trying to keep certain groups down, if we are ignoring the need to pay more attention to certain groups, to do the research in a different way, then we're still perpetuating the health disparities by ignoring. And so I think this gets tied to an ethical responsibility. I would argue a moral responsibility that we do our research in a way that truly is making it applicable and accessible for everyone to the best that we can. Understanding that there are challenges, understanding there are complications, knowing that not everyone who does research has to do health disparities research. That's not what I'm arguing. But I can say that whatever you're doing, you can try to make sure that you're including samples that are as diverse as possible. Making sure that when that's difficult to do, that you acknowledge that as a limitation in your studies, that you discuss how the findings might be different if you had different aspects of diversity that could be relevant. So just being mindful and incorporating that into the design or at least the discussion of research, I think still takes us a long way and helps us move forward much more than we have so far. So I kind of got ahead of my slides a little bit about what we can do because I already said some of this. But I do think that we can try to make small steps that add up to big steps when we all do them. So we can try to increase the diversity in our research samples. Again, that can be hard to do. We can do that through in trying to use more community engagement practices. There are researchers who specialize in this. There are people who provide consultation on this. How do you truly go into the community to engage people who normally are left out of research? It's not simple to just go into a neighborhood that has a certain SES or racial background and say, okay, we're here, come do our study. It doesn't work that way. So truly community-based research requires a give and take where you're providing services, you're providing information and resources, and not just taking in the sense of getting people to be in your studies. Even what I said is also an oversimplification. It's a field in and of itself to understand this, but I think we can draw on the people who've done this for years and learn how to engage different communities in our research. We can also do multi-site studies. I was talking about this earlier, that, you know, if you live in a place that doesn't have access to different groups that are relevant for your research questions, then collaborating with people in cities or in universities that have greater access is another way of being able to increase the diversity, knowing that you may not be able to do it where you are, but other people can work with you and collaborate with you to make that happen. We can also make better use of data that already exists. So I mentioned earlier that that my lab worked on a paper where we got a subsample of a publicly available data set. And we said, you know, this question is important to address in Black older adults. Let's just get the subsample and run the analyses. I didn't have to go and collect data on 170 Black older adults myself because I had access to it. And so I took advantage of what was already there. So we can do that. And we can also do things like data integration. So, you know, across different publicly available data sets, oftentimes there are common measures in different studies. So the demographics, some of the health information, some of the cognitive information, even some of the questionnaires might be the same or very similar across multiple studies. We can get larger samples of different racial and ethnic groups or other groups representing different aspects of diversity. We can increase the sample size if we get multiple studies and do the data integration techniques to be able to combine data sets. Now, I say that is not necessarily always simple. I've worked on this with different collaborators before, and it can be challenging. But again, I think it's worth the effort, and it's a way of using data that already exists, and particularly when it's difficult to collect some of that data ourselves. And then at the very least, like I said, the first two sets of recommendations I know can be challenging. I think we should try to seek to get closer to that. But in the meantime, the very least we can do is to make sure that we are very transparent about our data. I think sometimes people feel bad that they don't have a diverse sample, so they just don't report the demographics. And I think that that keeps us from being able to understand what the limitations of the study are. And I think it should always be reported. If it were up to me, every journal that has human research would require that the tables include race and ethnicity, sex, age, SES, and maybe even some other variables, and that be mandatory. Now, some things like SES, I know people don't always gather, but I don't know of any human researchers who don't have information about the other demographics. And I think that we should be reporting them, so that as readers, we can just evaluate, okay, this is a good study, but it's limited in this way. We should be able to have that as a way of interpreting what we're reading. And then, as I mentioned earlier, if nothing else, we can also talk about diversity in our discussion sections. Does that mean that every discussion section needs to have 5 paragraphs talking about every aspect of diversity? No, but when it's relevant, there are some aspects of what we do, some questions that we know that there's literature to suggest that there could be important differences across groups. And so just acknowledging that this could look different if our sample had more of X, Y, or Z group. That's something that can take all of a paragraph or half a paragraph, but it shows that consideration and it gives people ideas for future research. And that to me is one of the most important things we can do. Even when we can't do all of these grand ideas ourselves, we can inspire others to do it, and we can draw attention to the importance of it by making sure that we discuss it. So I will end with this quote that I absolutely love from a person who I very much respected and was very sad to see pass. But John Lewis said, if you see something that is not right, not fair, not just, you have a moral obligation to do something about it. So when I think about diversity and the research that we do, I do see it as something that's not right, not fair, not just, that we haven't done more. And so, as I said, I see it as ethical and moral prerogative on our part to do what we can about it. So I will leave you with that thought. I would like to acknowledge my lab. I have an amazing lab. I could not ask for better students, both past and current members. The pictures on the bottom are my current lab. I have a very big lab. At least for me, it's big, but they're fantastic. And acknowledge my funding sources. And thank you very much for your attention. I look forward to hearing your questions. You can contact me at my various email addresses that I have listed here. The Georgia State one is the best way to reach me. You can learn more about CerebroFit, my business that was mentioned. And if you'd like to hear more about the positive side, about some of the ways that we can promote brain health, my book that came out in March is written for the lay public to give guidance on how to live a brain healthy lifestyle. And if you scan the QR code, it'll take you to a page where you can order it from Amazon or from the publisher. So thank you again, and I look forward to answering your questions.
Thank you. Thank you so much. If we could stop sharing the screen at this time, visually rather, it would be easy just to see. There we go, easier to see each other. So far I only see one question in the Q&A, and it's not, it's more of a request. This person was asking if you might share the paper you were discussing related to exercise and brain health, the one that you mentioned here, Chuck.
Yes, I will email that to you and you can distribute it. And I'm happy to do that. I will do that once we finish up today.
While people are thinking about a question to ask, I certainly have one of my own, and want to just also comment actually to the last part, your last point that you were making. You were saying how we should all improve our reporting and, report race and gender, age range, and other factors. And I think that's absolutely true. Looking back, I think most of us were learned one way. We were trained to only show age and, maybe gender, and that's it. And I think it's become habit. I'm not sure if I'm signing that the right way, but yeah, it's just become habit where people have it's always been the way things have done. And I think my training, I was trained in Italy, and back then there was even less diversity in my country. And so for us, it was not top of mind at all. All the participants were always white, and that was just what we knew. And so for me, moving to America, that's not the same experience. Here, there's much more diversity. And also in terms of language background, so diversity of language background and life experience, just so many other diverse factors that I think it's really an important message that we need to do some introspection sometimes and identify where, quote unquote, our ways can be improved, And it might be a question that's added to our survey, or, something that's, identified further in, patient surveys, but it doesn't have to be something complicated. I think you want to focus on something that you can select from a specific group, or your data can say something about a specific group. And then add to a new perspective, or not add a new perspective rather to the data.
I 100% agree with all of that. And it's interesting when you were just talking about how we do what we've been taught, because even though I'm one of the people sounding the alarm right now that we need to do more, I am guilty of the things that I'm criticizing the field for. I went back and looked at some of the first few papers that I wrote, and I was so angry with myself because I realized that I did not consistently report some of these factors either. And I think it's a legitimate point. It's not an excuse. We do what we were taught. We do what our mentors did. We do what we see in papers. And it takes a while sometimes to sort of wake up and realize, hey, this should be done differently. Let's challenge like what's been done. Like you said, let's introspect. It's a difficult thing to do, but an important thing to do. So I really appreciate your comments. Thank you.
Hi, I do have a question. And then I'll turn it back over to the students if they have any other questions. Would you mind just expanding a little bit on how intersectionality, how it relates to your work? So a lot of our audience today is, we will study groups who are deaf and hard of hearing. So we know a lot about that. And that's something we're comfortable with. And that's a minority group, but what happens often is it's a problem with the size of the sample and who we're studying. It ends up being a small data set. We can collect information on a lot of deaf and hard of hearing people, but then when you analyze it deeper, you see there's, not a lot of racial diversity, not a lot of diversity amongst SES levels, and it becomes a true challenge to make sure that you're covering all the bases and analyzing the true variability within the human experience. But, we're gathering data from already a small group, of deaf and hard of hearing people. So I don't know if you could just expand a little bit about that. I know you've thought about that before.
Yeah, absolutely. So you bring up such a good and important point that as we get into groups that are already more rare, it becomes extra hard to try to then also have representation of other areas. So getting at the intersectionality piece is really, really hard. So first, let me say, I know that there's not a single study that can address everything. And so if you're studying a population that is already unique, already marginalized, like a group that's deaf and hard of hearing, you're already doing something incredibly important and addressing gaps in the literature. And you can't necessarily then also have all other aspects of diversity represented. That doesn't mean that you don't try to diversify. It just means that it's understandable that that might be difficult. What might happen is that as different researchers who are doing studies in the same population make their data available to each other to combine later, then eventually, it might take some time, but eventually then you can start to look at subgroups within there because you have the power of multiple studies combined together. That takes time, but it is one of the things that's really good about the push towards open access for our data, because it allows for those kinds of things to be done after the fact later on. So I think that hopefully addresses most of what you were getting at. I will say for me, it can get difficult because even for looking at vascular depression, and especially because I do exercise research, I'm trying to get Black older adults with vascular disease and depression who are willing to exercise for 4 months. Oh my gosh. Yes, the sample does get smaller and smaller. And I've learned to be realistic about realistic recruitment goals, and understanding that even small pilot studies can be valuable, and that we'll keep working on how to better engage that really needed and vulnerable population, but it's challenging. And if I were to add any other aspects of diversity into that, then I think I would never get any research done. So there is something about picking pieces that you can address with each study, knowing that not one study can address all of it. But if we all try to get one piece of it, then we'll eventually cover different groups, if that makes sense.
Yeah, thank you. I really appreciate that response because it's complicated, and there's not a simple answer or solution. So now I'll wait just for a minute. We'll go through an awkward moment of silence, but I did wanted to ask the students if anyone wanted to turn on their camera and ask a question, or even put your question in the chat. Oh, we see a hand that's been raised. Okay, I will allow that person to turn on. I think I have to make them a panelist so that they can turn on their video. And hello. First, thank you so much for your talk today. It was really, the translational implications are amazing. So thank you. Just to name a historical context of this kind of research, what do you think the current climate, or the recent climate, I guess, how does that recent climate impact your research looking at it moving forward? Your generation, our generation, as you mentioned, they're going to look different. So that's a question I'd like to ask.
Yes, thank you for that question. I'm really glad you enjoyed the talk. Oh, I could be here all day if I talked about all the ways that the recent climate impacts the work in ways that are good and in some ways challenging, to be honest. I appreciate that right now there is more of a focus on inequities in society across different groups. We, of course, in 2020 in particular, it was a big period of racial reckoning, but I think that also reminded people that, hey, there's a lot of vulnerable groups that really have been ignored, marginalized, and harmed in various ways. So in some ways, I think the climate is making it more feasible to do the work. NIH is putting more funding towards health disparities work and trying to do more to fund people from diverse backgrounds, research that's on different vulnerable groups. I think all of that's wonderful. What's interesting, and something that I probably should have anticipated, but I did not, is that, my lab has been starting to get some pushback from people in the community when we recruit for studies in Black older adults. I had a really upsetting, to say the least, email that one of my graduate students received. So this is a Black female grad student who sent an email to a community center about our study looking at exercise for vascular depression in Black older adults. She briefly mentioned what we're doing, that we're trying to study this vulnerable group. We're recruiting people who identify as Black over the age of 65, et cetera. So the director of marketing for that community center writes back to her. He didn't know that I was going to see the email, saying, we will not tolerate racism that we are offended that you would not include all of our residents. There's no reason to think that race would affect anything that you're looking at. And all these accusations that, number one, were unfounded, number two, were incredibly offensive. My poor graduate student was just, devastated. She was just, imagine as a student having someone accuse you of these kinds of things when you're trying to do research to actually help a really vulnerable group. So I got that email and I had to calm down. I had to take a couple of hours before replying because she forwarded it to me because I didn't want to be unprofessional, but I was really pretty livid. It felt to me like one of those situations where in some ways the current climate has led some people to feel protective of the status quo. And so my thought was, well, people weren't complaining when studies were only including non-Hispanic whites. Why are we complaining when we're trying to target certain groups? So I ended up sending a professional, but still very stern email saying that I did not appreciate the accusation to my graduate student who in no ways was being racist. I sent a quote from Merriam-Webster from the dictionary about what racism is and said, this is not racism. This is targeting a vulnerable group in the same way that since white older men are more at risk for suicide, lots of studies focus on white older men. So if we have a different condition that has other groups that are at risk, we study them. And sent him actually both of the papers that I sent you all. So I tried to provide education and then spent some time processing it with my student and the rest of the lab because we realized there are some ways in which the focus now in our current climate on marginalized groups for people who are not marginalized can feel threatening and lead to pushback. And that's unfortunate, but it's what happened. And so that is sort of a long story to say, I think there's good and bad about the current climate when it comes to the work we do. I will do it regardless and push through with the negative that comes up because I feel it's really important.
Thank you. Unfortunately, the person who had raised their hand before I don't see their name on the participant list anymore. So that's unfortunate. But while our students may be thinking of, I don't know, we have about a minute or two left for questions or a comment, I just wanted to make a statement that the government genetics, in terms of, the government agencies, pardon the interpreter, who are providing funding for studies and research, those government agencies are focusing on diversity and have a clearer emphasis. They are looking for more of an effort to be placed on diversity for projects that they fund. But the problem is, when you have a panel, for example, an individual review panel, if the panel isn't focused on diversity, if they are sort of ignoring or not paying attention to that fact, that's where we can strengthen our focus, I think. And, if the program office doesn't tell the panel, hey, listen, you have to be critiquing that part, then often people won't. And, the emphasis on diversity is not there to awarded entities. And so when we want to make our research more broad and more diverse, we also have to have the budget for it. So being diverse in our studies requires more money because you have to send more students, you have to do more outreach. Travel expenses are more expensive because, as you had said, at your former university, there wasn't a lot of diversity, which means if you want to collect data on a certain group of people and you don't have enough, then you're going to have to travel to another city and perhaps stay in that city for, so there's hotel fees and, a lot of different costs that accumulate. And that's where I think that we could still You know, there's still that part of we're falling back into our old habits where we're prioritizing money. You know, we want money for the research and the experiment itself, but there's not enough money in the recruitment, invested in recruitment. And so I think we should spend more money on recruitment, honestly, than the actual experiment piece. Maybe if we flipped it that way, we'd see some positive results.
I love that. What you said at the end, please, let's do that. It's so true. I 100% agree with everything that you said. I think it's going to be slow progress to get things to change in that way. I hope we're headed in the right direction, that people will start to, that review panels will start to have that top of mind as opposed to an afterthought or something that's dismissed, that there will be more leniency with budgets, that individual researchers will change how they proportion the money in their budgets. And I know we're going to get low on time, but I did want to just quickly add that I also think that something has to change in academia where the quality and type of research gets focused on more than numbers, because people going up, for a tenure and promotion are oftentimes thinking, I got to get publications out. I need what's going to be quick and cheap. And there's no incentive for doing the hard work that it takes to do inclusive research. So until the people who evaluate us and decide if we keep our jobs have that same value system, there's going to be a limit in how far we'll be able to go with these efforts. So thank you for that.
Yeah, those are golden words.
Thank you.
All right, well, time is up for this afternoon. And when it's a good time always goes quickly. That's what I say when you're having fun. So thank you, everyone. Thank you so much, Dr. Dotson, for joining us today. And thank you to our audience. And, we saw so many people here, and unfortunately, not all of our students, not all of our faculty, but we even saw people from other departments join and attend today, and some familiar faces. So thank you so much.
Thank you again for having me. It was a great pleasure.
All right, I'm just going to stay on. If anyone has any questions for me, you can turn on, but I'll just stay until everybody leaves, then close down the platform. Thank you. Bye-bye.
Thank you.
So nice to meet you.